Now we will learn how to change the media in the T75 cell flasks. First, aspirate out all of the old cell media in each flask. Avoid touching the side that has the cells on it. Next, add 12 milliliters of cell media into each cell flask. The amount of media is dependent on the size of the flask. For this, we are using flasks that are 75 centimeters squared, so we use 12 mils of media. Then place into the 37 degrees Celsius incubator. After every step with the cell flasks, be sure to check their confluency under the microscope located next to the water bath. Here are some examples of different confluency of the cells. Confluency defines the amount of space that the cells take up on the bottom of the flask. This is an extremely qualitative way to measure the cells, and is just for personal reference. We will now learn how to freeze cells. You will need phosphate buffered saline, also known as PBS, 0.25% trypsin. This can be found in the -80 degree. In order to open the flasks, open the top one first, followed by the second. This will avoid moving over them and possibly contaminating them. Aspirate out all of the media in both flasks. Do this by tipping them to the side and aspirating on the side with no cells on it. Rinse with 5 milliliters of PBS in both flasks. This is dependent on the size of the flask. Our flasks are 75 centimeters squared, so we use 5 milliliters to completely cover the bottom. Slowly rinse the bottom of the flask by oscillating back and forth with the PBS inside. Aspirate out the PBS. Add 5 mils of PBS again and repeat the rinsing process for a total of 2 times. Add 3 mils of 0.25% trypsin to each flask. Before adding, mix the trypsin with a pipette because it was previously frozen and some of the particles could be stuck at the bottom. In order to mix the trypsin, pipette some of it up into the pipette and then pipette it back down into the solution. The trypsin causes the cells to detach from the bottom of the flask. The amount of trypsin is dependent on the size of the cell flask. With the 75 centimeters squared cell flask we use 3 mils of trypsin. Oscillate the cell flask from side to side to make sure that the trypsin completely covers the bottom of the flask. Now place the cells into the 37 degree Celsius incubator. The incubator will speed up the trypsin process. Leave the cells in the incubator for roughly 3 minutes and then examine under a microscope. The cells will look like little round balls when they are all detached. After incubating, add 3 mils of media into each cell flask. This will prevent the trypsin from harming the cells further. When removing the cells from the flask, pipette some of the solution and inject it back down onto the bottom of the flask. Find a systematic approach to this. Repeat this step several times. Once done, add all cells to a 15 milliliter centrifuge tube. Next, centrifuge at 180 Gs for 7 minutes. Make sure that the tubes have the same volume when centrifuging, and place on opposite sides of the centrifuge to balance it out. For the next step, you will need an epitube. After centrifuging, you should be able to see a small white cell pellet at the bottom of the centrifuge tube. These are all of the cells from our cell flasks. Slowly aspirate out the media and trypsin mix. Be very careful of the cell pellet at the bottom. Tipping the centrifuge tube as much as possible makes this easier. Add 1 ml of PBS into the cell pellet centrifuge tube. Mix the cell pellet in with the PBS by slowly pipetting up and down. Now add 9 milliliters of PBS into the PBS and cell mixture. Mix this thoroughly using the pipetter. Now add 200 microliters of the cell PBS mixture into the epitube. This will be used for the cell counter. Turn on the cell counter by using the black button on the back. To use the cell counter, plug in the sensor by matching the gold side on the sensor and the prongs on the counter. Press the plunger down and then submerge the sensor in the cell solution. Do not let the sensor touch the bottom of the epitube. Slowly release the plunger until the screen says that the sample is loaded. Once the sample is loaded, the screen will show this. Remove the sensor to continue to the data. The histogram shown represents collections of cells of different diameter. The given concentration, shown at the bottom, is calculated from the peak of the histogram. Knowing the diameter of cells, in this case fibroblasts are 10 to 15 micrometers in diameter, you can tell if this accurately represents your sample. If there are any bimodal or skewed distribution in the histogram, this could mean contamination of your cell solution. Cells are frozen in groups of 1 million cells. In this group of cells, we had 1.6 million. We can set up a proportion to determine how many milliliters of the cell solution we need in order to get 1 million cells. Using the calculations on screen, we find that 6.25 milliliters are needed of the cell solution. Now I will now transfer out 3.75 milliliters into a separate container in order to get 6.25 milliliters in the original centrifuge tube. This centrifuge tube will contain 1 million cells, and we will freeze all of those cells. This centrifuge tube will need to be centrifuged at 180 Gs for 7 minutes. After done centrifuging, aspirate out the PBS media. This next step will require HDF freeze media. HDF freeze media is 90% fetal bovine serum and 10% DMSO. Add 1 milliliter of freeze media to the cells. We use 1 milliliter per 1 million cells. Mix up the cell pellet inside the freeze media. Add the 1 milliliter of cells and freeze media into the cryotube. Label the cryotube with the cell line, how many cells, the passage number, your initials, and the year that they were frozen. To freeze the cells, place into the refrigerator for 10 minutes. Then into the -20°C freezer for 10 minutes. And then into the -80 freezer permanently. Place into one of the boxes.